Good afternoon, everyone, and welcome to today's session. As usual, we'll wait for 5 minutes so that people can join in. Okay? I guess we can start our today's session. So, unfortunately, the last class which we were discussing, only half hour of it was recorded, and due to some issues. So, I might repeat some of the things. So, for you who were present in the class, it for starting 15 minutes will be quite repetitive, but I hope you will keep the patience. As I have to make sure that this time it is being recorded and everything is on fleek. Let me just check that. Yes, so I'll just start my lecture for today. So yeah, so just a quick recap whatever we have discussed in the last class, and then we can start from wherever we have left. So we were discussing about the secondary structures, then we started discussing about certain properties of secondary structures and why do we actually need secondary structures and how they are there present, like how secondary structures are present there, right? So for the beginning we were talking about that protein structures are stabilized by non-covalent interactions and forces, which basically means that though the covalent bonds are the strongest type of bonds, but the stability of the overall protein does not completely depend upon the covalent bond. It is more towards the non-covalent interactions. Cause formation of a thermodynamically favorable structure depends on the influences of the hydrophobic effects, hydrogen bonds, ionic interactions, and van der Waals forces. Natural protein structures are constrained by peptide bonds whose configurations can be described by the dihedral angles. Protein segments can adopt regular secondary structures such as the helix and the conformation. These structures are defined by particular values of phi, and their formation is impacted by the amino acid composition of the segment. All of the phi angles and the values for a given protein structure can be visualized using Ramachandran plot. And from here we digressed towards the Ramachandran plot. Let me bring that again for you guys. Yeah, just give me 1 minute.
Okay, this is an excellent.
We'll do this quickly.
Hello everyone,
here.
myself Zeel
Yeah.
Bhatt and welcome to my channel Zeel's Pharmacy Tutorial. And today we are going to study about Ramachandran plot. I am making this video on the request of one of my comments. It is a way to visualize dihedral angles psi against phi of amino acid residues in protein structure. In the simple words, it is a way by which we could identify secondary structure of proteins depending upon the rotation or degree of freedom of dihedral angles psi and phi. Secondary structure of protein could be alpha helix, beta sheet or loop structure. So now, let us study about characteristics of Ramachandran plot. First, it contains 4 quadrants. So the Ramachandran plot is divided into 4 quadrants. First quadrant, second quadrant, third quadrant and fourth quadrant. Then second, y-axis will be psi angle and x-axis will be phi angle. So in the Ramachandran plot, this is psi angle on y-axis and phi angle on x-axis. The angles range from plus 180 degree to minus 180 degree on both the axes. So what are psi and phi angles? In this structure, oxygen is more electronegative than carbon. Hence it pulls electron pair towards it, making carbon positive. As the result, the lone pair of nitrogen creates partial double bond. Thus, it makes this bond rigid. The bond between C alpha and N is called phi bond, which is free to rotate. Similarly, in the case of C alpha C, the bond is called psi bond, which is also free to rotate. So the torsional angles or dihedral angles formed between C alpha and N is called phi angle, and the torsional or the dihedral angle formed between C alpha and C is called psi angle. Now the rotation mainly depends upon this R. If this R is simple, For example, as in glycine, it is H. The rotation is more. Similarly, if R is of proline, which has a complex structure and is bulkier group, the rotation is very difficult. Then third, the finite position in the plot will indicate the finite secondary structure of protein. This region in first quadrant indicates antiparallel beta sheets. Then this region in the first quadrant indicates parallel beta sheets. Similarly, in the second quadrant, this region indicates left-handed alpha helix, and in the third quadrant, this region indicates right-handed alpha helix. And the last fourth, the plot contains both allowed and disallowed region. In computing Ramachandran plot, atoms are treated as hard spheres where dimensions corresponds to their van der Waals radii. Any angle that results into the collision of the spheres are regarded as sterically unfavorable, and hence such conformation are not allowed. Based on this principle, the graph is divided into three regions. First is the most allowed region, then second is allowed region, and the third is disallowed region. Most allowed regions do not have any steric hindrance. Allowed region corresponds to the conformations that could be possible if slightly shorter van der Waals radii are used in calculation. Disallowed region corresponds to the conformation that are sterically unfavorable, and this is due to the presence of the bulkier group. Here, red color regions are most allowed region, then yellow color regions are allowed region, and white color regions are disallowed region. Thank you.
Right, so it was a very brief about the Ramachandran plot. If you guys want, we can have a separate maybe 10 to 15 minute discussion specifically for Ramachandran plot, and we can look into how it was determined, how it is used even till today to know whether the protein structure whichever we have determined by cryo-EM or X-ray crystallography is actually a protein which exists, right? So we still validate our protein structure by plotting them on the making their Ramachandran plot. Okay, so the tertiary structure describes the well-defined three-dimensional fold adopted by a protein. So protein structures are often built by the combinatorial use of common protein folds or motifs. Quaternary structure describes the interactions between components of a multi-subunit assembly. Tertiary structure is determined by amino acid sequence. Even though protein folding is complex, some denatured proteins can spontaneously refold into their active conformation based only on the chemical properties of their constituent amino acids. Cellular proteosis involves numerous pathways that regulate the folding, unfolding, and degradation of proteins. Many human diseases arise from protein misfolding and defects in proteostasis. So basically, if we know the sequence of a protein, that is why knowing the sequence of a protein was such a milestone in structural biology. Because if we know the sequence of a protein, we know the chemical properties of each amino acid residue at the physiological pH and temperature, right? So we can presume and we can guess what sort of structure or what sort of function that protein might be performing, or what structure finally it will take. So based upon our basic biochemistry, basic chemistry, we can actually get to an idea about the function or the structure of a protein whose structure might not yet be available. Okay, the three-dimensional structures of proteins can be defined. So structural biologists use a variety of instruments and computational methods to solve biomolecular structures. The choice of method may depend on factors such as the size of the protein being studied, its properties, or the desired resolution of the final structure. The possible conformations of a protein or protein segment include any structural state it can achieve without breaking covalent bonds, right? Because if you break the covalent bond, the polypeptide will break and hence it won't be a protein anymore, right? A change in conformation could occur, for example, by rotation about single bonds, as just now we have seen in the video that both phi and psi bonds have the ability to rotate and move. right? But they are not breaking. It's just the spatial arrangement of the groups around these bonds are changing, and they can change, right? However, of the many conformations that are theoretically possible in a protein containing hundreds of single bonds, one or a few generally predominate under biological conditions. Yeah, so this is a very important line, because if theoretically we are saying that single bonds, both phi and psi, can freely rotate, right, without breaking, indicating that there will be infinite number of possibilities. There will be infinite number of possibilities or arrangements, right? Theoretically it will be possible, hence infinite number of conformations should exist for the protein also, correct? But this is not the case. So why this is not the case? Because there are many orientations which are not at all feasible under the physiological conditions. Hence we only see predomination or domination of few of the states which are present at the biological conditions. Though infinite number of possibilities are present, yet due to the biological conditions there are restrictions to the available orientations of the functional groups around the single bonds. Okay. The need for multiple stable conformations reflects the change that must take place in most proteins as they bind to other molecules or catalyzed reactions. The conformations existing under a given set of conditions are usually the ones that are thermodynamically the most stable, that is, having the lowest free energy. Proteins in any of their functional folded conformations are often called native proteins. Okay. The stability is the tendency of a protein to maintain a native conformation. Native proteins are only marginally stable. The delta G separating the folded and unfolded states in typical proteins under physiological conditions is in the range of 5 to 65 kJ per mole. A given polypeptide chain can theoretically assume countless conformations, and as a result, the unfolded state of a protein is characterized by a high degree of conformational entropy. This entropy, along with the hydrogen bonding interactions of many groups in the polypeptide chain with the solvent, that is water, tends to maintain the unfolded state. The chemical interaction that counteract these effects and stabilize the native conformation include disulfide bonds and the weak non-covalent interaction and forces, which we're gonna see in the coming class, in the coming slides. But before that, we'll quickly see these links so that we can have a better understanding of our 3D structural conformation and how they are determined.
Protein called lysozyme. It represents the order of nature, and I feel awed when I look at it. It's an enzyme that's in our tears and saliva and mucus, and it helps us fight bacteria. And knowing this three-dimensional structure helps us to understand the mechanism of action of this enzyme, and therefore how it helps fight bacteria in our bodies. Unfortunately, the size of the molecule is such that it's far too small for us to see under a light microscope or with our naked eye, because the wavelength of light is much, much larger than the size of this tiny molecule. For that reason, we have to use radiation that's much smaller in wavelength, and we use X-rays to look at these molecules. These are protein crystals. Locked within each crystal are millions of protein molecules, all arranged in an ordered grid-like structure. By firing X-rays at these and measuring how they scatter, we can work out the molecular structure of nearly any crystallized sample. It's through this method, known as X-ray crystallography, that some of the most important biological structures have been obtained, from the double helix of DNA to numerous proteins, vitamins, and drugs. But getting from a crystal to something like this, the structure, is not at all trivial, and it can take a long time to grow a suitable crystal. Now we're in the protein production and purification lab, because before we can set up crystallization trials, we need to produce enough protein for that process. We do this by genetically modifying E. coli bacteria, which then acts as a little factory to produce our protein in large flasks of broth, which we incubate. Once that's happened, we break open the cells, extract the protein, and then purify it, ready for the crystallization process. So why do we need to grow crystals of our protein molecules before we can shine X-rays at them and try and find the three-dimensional structure of them? Well, if we imagine that this string of beads is a protein molecule made up of 20 different amino acids, different colour of beads that are found in nature, this folds up in a very complex, complicated manner in the three-dimensional shape like this. And if we look at one of these, true biological molecule here, a real one, what this metal model represents is a string through the beads, and you can see it starts at this end and it follows a pretty tortuous path going around here that you'd never imagine when you just look at the string of amino acids, and the other end of it comes out here. Now it turns out that if I take a tube here with my protein in it that I've purified and prepared, there's millions and millions of protein molecules in there. And if I shine X-rays at it, the X-rays will scatter off in all sorts of random directions, and I won't get any information about the shape of the molecule within the tube. However, if I can get the protein molecules to line up in an ordered array, such as in a crystal, where they're all lined up in the same orientation, when the X-rays scatter from the crystal, then I can get enough information, the signal is strong enough for me to get the three-dimensional structure of the protein. We've now come down to the crystallization lab to look at how we crystallize protein. In this Petri dish, I've got some supersaturated sodium acetate, and that means that there's so many molecules crowded in this solution, it's almost not holding the molecules, and it wants to solidify. And if I hit it with this spatula here, you can see that it crystallizes. We get a fantastic pattern as it crystallizes across the dish. Essentially, this is what we try to do with our proteins, which is to produce a supersaturated solution of the protein, and we dehydrate in a very controlled manner. The proteins we work on here, unfortunately, can be sometimes really difficult to crystallize. So we load small volumes of the protein into trays like this with different additives, but we have robots that help us do that by pipetting small volumes into these trays.
I guess you guys can see all this later, as I've already discussed about them in the last
Once we have the tray with the protein and additives in, we won't get any information.
Yeah. So I'll quickly stop sharing it so that we can go back to our slides and cover some new topic. Yeah, and this was with respect to the cryo-EM technique, which is another technique used for structure determination of the proteins. So this is the basic flow of how the cryo-EM process takes place. So you'll have a sample preparation, you'll overexpress your protein in a sample, and then you'll just put it on certain grids which are there with the microscope, and then you'll image it right at very low temperature then you'll do the data collection, then there are certain pre-processing steps, then you'll try to map these structures, these reconstructions, and then you fit the reconstruction to a model, and then at the end of the day you can do your analysis and assign all the 3D coordinates of to each and every atom of the protein. On the other hand, if we look at the workflow of X-ray crystallography, again your protein of interest is purified, and before the crystallization, right, as the name suggests, you need to crystallize it first. Then second is the crystal plate setup. In this, basically the protein and the crystallization mix are added to a plate. It can be of anything, it completely depends upon your setup. Then it's the crystallization process. The protein crystallizes under unique condition, so that there is no like non-uniform crystals or all those kind of technical difficulties. Then at the end of the day, we protein crystals are shot with X-rays to produce a diffraction pattern unique to the protein structure, because every atom and every residue in that crystallized mixture will diffract differently. Okay, and then last but not the least, we have phasing, model building, and refinements. So in this, using the diffraction pattern and specialized softwares, researchers and scientists are able to determine the structure, the 3D structure of a protein. There are other ways also, but these two techniques are the one which are highly used for determining the structure. As it is a course related to the membrane proteins, you guys already know that structure determination of membrane proteins is quite difficult even with these techniques, but still, as the technology development is proceeding, we can hope that there will be more and more membrane protein structures also available in the protein database. Okay, so we were talking about the forces which might disbalance the structure of a protein, right? So some of them can be a covalent disulfide bond. They are strong, but they are also uncommon, right? Because why they are uncommon? Because if you remember, we have only 2 sulfur containing amino acids, out of which only one amino acid, that is cysteine, has the ability to make disulfide bond. Okay, the environment within most cells is highly reducing due to high concentrations of reductants such as glutathione, and most sulfur hydrides will remain in the reduced state. Outside the cell, the environment is often more oxidizing, and disulfide formation is more likely to occur in such conditions. In eukaryotes, disulfide bonds are found primarily in secreted extracellular protein, for example, the hormone insulin. Disulfide bonds are also uncommon in bacterial proteins. However, thermophilic bacteria as well as the archaea typically have many proteins with stabilizing disulfide bonds. This is presumably an adaptation to life at very, very high temperatures. For all proteins of all organisms, weak interactions are especially important in the folding of polypeptide chains into their secondary and tertiary structures. The association of multiple polypeptides to form quaternary structures also relies on these weak interactions. About 200 to 460 kilojoule per mole are required to break a single covalent bond, right? So it's a huge energy requirement, whereas weak interactions can be disrupted by a mere 0.4 to 30 kilojoule per mole of energy. Individual covalent bonds such as disulfide bonds linking separate parts of a single polypeptide chain are clearly more stronger than individual weak interactions. Yet, because they are also numerous, the weak interaction predominates as a stabilizing protein structure. So this is quite important, because we know that a covalent bond requires almost this kilojoule per mole of energy, which means wherever the covalent bond is present, the structure will be stable. And if we look at the energy required to destabilize a non-covalent interaction, it is merely 0.4 to 30 kilojoule per mole of energy, which is somewhat easily accessible within the biological system, right? But though individual weak interaction or non-covalent interaction is weaker, but they are numerous in number, right? So the weak interaction predominates as a stabilizing force in protein structures. In general, the protein conformation with the lowest free energy, that is the most stable conformation, is the one with the maximum number of weak interactions. I want you guys to remember this very well, that the protein conformation with the lowest free energy, it is a very general thing, right? Because in the biological system, for the stability purpose, lower the energy, more stable the structure or the system is, right? So in with respect to proteins, the lowest energy structure or the conformation which is highly stable will be the one which has a maximum number of weak interactions. Hence, whenever proteins amino acids are connected via peptide bond, that is the peptide chain, they will try to adopt a conformation which will maximize the weak interactions, right, and minimize the total energy of the formation of that particular 3D structure of a protein. Okay, the stability of a protein is let me get back to my the stability of a protein is not simply the sum of the free energies or formation of the many weak interactions within it. For every hydrogen bond formed in a protein during folding, a hydrogen bond of similar strength between the same group and water was broken. The net stability contributed by a given hydrogen bonds, or the difference in free energies of the folded and unfolded states, may be close to zero. Ionic interactions may be either stabilizing or destabilizing. We must therefore look elsewhere to understand why a particular native conformation is favored. So this is not as straightforward as we are trying to depict it, right? So we are saying we can maximize the non-covalent interactions, which will minimize the total energy, and hence that will be the most stable structure. But if you look at the biological conditions, if a polypeptide chain is formed, it will be in the aqueous environment. There are amino acids which can form hydrogen bond with the cytosol itself. So what energy or what motivation will these polypeptide chains should have so that they can break the hydrogen bonds which are making them stable in the water to come out of it and make hydrogen bonds within themselves for the 3D structure, right? So this cannot be the only driving force for the stability of a protein structure. There has to be something else also, so that proteins can come from unfolded state to folded state. Okay, so till now we were discussing about the alpha helix type of secondary structure. So another major component of secondary structures is called pleated sheets or beta pleated sheets. Excuse me. Yeah. So in 1951, again Pauling, they gave a paper titled "The Pleated Sheet: A New Layer Configuration of the Polypeptide Chains." For many years, it has been assumed that in silk fibroin, stretched hair, and muscle, the other proteins with the beta keratin structure, the polypeptide chains are extended to nearly their maximum length, about 3.6 angstrom per residue. And during the last decade, it has been assumed also that the chains form lateral hydrogen bonds with adjacent chains, which have the opposite orientation. A hydrogen bonded layer of this sort is represented in the diagram, which is this. So these are the beta sheets, right? To be specific, these are the anti-parallel beta sheets. As you can see, first N terminal is here, second N terminal is here, third N terminal is here, fourth N terminal is here. So these sheets, they form intermolecular hydrogen bonds, and the polypeptide chain runs in the anti-parallel direction, right? There are parallel beta sheets also. So if you look at the parallel beta sheets, you can clearly see that their N terminals are in the same direction. but you can clearly see that there is some difference in the way that they make the hydrogen bond. In parallel beta sheets, the hydrogen bonds are pretty more extended, while in anti-parallel beta sheets, they are more ordered and oriented. Okay, so we'll quickly look at the beta sheets so that we understand more about them and their basic properties. Okay.
Hello everyone, in this video we'll talk about the secondary structures of protein, and in that context we would talk about beta plated sheets. Previously we have talked about alpha helices. If you missed that video, go check that video and the link is in the I button. So protein structure can be divided into different structural levels, starting from primary structure, then secondary structure, tertiary and quaternary structure. And now we are focusing on the secondary structural features. Among the secondary structural features, there could be alpha helices, there could be beta turns, there could be beta pleated sheet. The center stage of this video would deal with beta pleated sheet. Now, beta pleated sheet is a region of peptide chain where the amino acids are linked with each other in a specific orientation in form of hydrogen bonding, and this kind of gives like a sheet-like appearance in a 3D space, and that is why the name is beta pleated sheet. So let's look at the structural features of this. So the NH groups of the backbone actually forms hydrogen bonds with the carbonyl group from the other amino acids. So roughly it looks like this. Now the biggest difference between alpha helices and beta plated sheet is the distance between adjacent amino acid, which is 1.5 Armstrong in alpha helices, whereas it is 3.5 Armstrong in beta pleated sheet. So in terms of steric hindrance, beta pleated sheet structures experience less steric constraint compared to alpha helices. Now, beta plated sheet can be organized into two different orientations, parallel and anti-parallel. In this particular orientation, you can understand the beta plated sheet has all the hydrogen bonds in a particular plane, right? In a planar fashion. And all the amino acid R groups are protruding out from the plane either upwards or downwards. That itself is reducing the steric constraint, right? Now let me tell you, due to these kind of structural organization, tryptophan, tyrosine, phenylalanine, these kind of bulky side chain containing amino acid can be a part of beta pleated sheet. But this is not a stereotype. There are many other structures where beta pleated sheet generally prefers smaller side chain containing amino acid as well. And probably due to the structural feature of beta plated sheet, these kind of bulky groups can fit. Now, if we look at the beta plated sheet in a Ramachandran plot, we would see the phi and psi angle would be roughly minus 120 degree and plus 115 degree. Roughly they fall into this particular region. Both parallel and anti-parallel sheets fall into this region. Now the question is, where do we find the beta pleated sheets? It turns out many protein contains both alpha helices or beta plated sheet as their secondary structures, but the proportion is different. Some proteins might be enriched in beta plated sheet while some others are enriched in alpha helices. Just to give you an example, the silk or fibroin, this particular protein, is very much enriched in beta pleated sheet. Now, the supramolecular association of beta plated sheet has been implicated in neurodegenerative diseases like Alzheimer's, where it can form aggregation. So beta plated sheet, their interaction with each other is very important in context of disease. Now let me tell you that how structural features of proteins can be studied using experimental techniques, like how we can experimentally determine the changes at the level of beta plated sheet or alpha helices. Techniques like X-ray crystallography or circular dichroism spectroscopy could be useful to study these features. So if you want to learn about these specific techniques, the link would be provided in the I button. Now here, let me tell you about circular dichroism spectroscopy. Looking at the CD signature, you can understand whether that particular protein is majorly constituted with alpha helix or beta plated sheet. Let's take one example. Look at this particular dotted line. This is the protein signature or CD signature of a particular protein. Now comparing with known signature, you can kind of understand this protein would be majorly made up of alpha helices. Then coming to the next particular example, where we see a stress like this. This particular protein looks like it is going to be majorly constituted of beta plated sheet. Apart from these techniques, cryo-electron microscopy can also provide us with the structural information about a protein. So, in this video, what we have learned is the structure of beta plated sheet, where we can find them, etc. But these
Right, so this is another major secondary structure, that is the beta sheet. And even in the beta sheet, there are multiple orientations like parallel beta sheet, anti-parallel beta sheet. But if we look at our physiological proteins, the proteins for which the structures are already there, so it won't be always that the protein will always be a alpha helix or it will always be a beta sheet. So there are 3D orientations and 3D motifs or domains which actually have a combination of alpha helix, parallel beta sheet, anti-parallel beta sheet. So there are some determined motifs in the biological system which have a unique function and a structure associated with them. Okay, so in the next class we're gonna look into a few such motifs and what their function is, and this information actually help us in determining the function of a protein, for which for example, if it's a new protein and we don't know its function, so if we know that this is a protein and it has that X motif, for example, and from our experiments and the information and the knowledge which we have with respect to that motif, we can assume that if that motif is present in this new protein will also perform similar function. Okay, so knowing these motifs, knowing the functions, how they are doing, the certain characteristics of these motifs help us in determining the function of a newly formed protein or newly discovered protein. Okay, so we will see all these things in the next class, which will be on Tuesday as usual. Okay, thank you so much for your attention and hope you guys are learning, and if you have any queries, feel free to drop me a mail. Thank you so much.